Hello everyone, and thanks for joining me today on Study FA with me. My name is Arzo, and I will be your guide to getting through chapters in the first aid with my own notes and mnemonics on how to understand and memorize the high-yield topics that are asked on the NBME exams and the USMLE STEP exams. Remember that first aid is a revision book and not the only source needed to get you through the exams. So with that said, we will begin our first video on microbiology. So we'll start with bacterias with exotoxins. 4 bacterias inhibit protein synthesis. This is Corynebacterium diphtheria, Pseudomonas aeruginosa, Shigella, and E.coli. The way I remember these bacterias is DASE, D-A-S-E. So diphtheria, aeruginosa, Shigella, and E.coli. Diphtheria contains diphtheriae toxin, Pseudomonas aeruginosa contains exotoxin A, Shigella has Shigatoxin, and E. coli has Shigella-like toxin. So because Shigella and E. coli have almost the same toxin, you can assume that the side effect is going to be very similar. To inactivate translation elongation factor 2. So that is going to be Corynebacterium diphtheria and Pseudomonas aeruginosa. The mechanism is that both of these inactivate the translation elongation factor 2 in eukaryotic cells. Corynebacterium diphtheria, the toxin is that it's a DNA beta prophage that encodes for your diphtheria toxin, and this is identified by your ELIC or your ELISA test. ELISA is now a gold standard. It's also cheaper, but ELIC is a gel test. You will see black-gray colonies of club-shaped rods arranged in V or L-shaped on your cysteine tellurite agar. Sometimes they will tell you that it's black-gray colonies, and you're supposed to assume or identify that this is diphtheria toxin. On Loeffler media, we will see metachromatic blue and red granules. The mechanism of the bacteria is that it will inhibit eukaryotic elongation factor 2 via ADP ribosylation. It's a gram-positive rod that is identical to normal flora organism of the oropharynx, known as diphtherioids. So this is a process that undergoes lysogeny, which we will talk about next. So the ELIC test. The ELIC test involves diffusion in gel and a precipitation reaction that occurs when the toxin or antigen produced by the pathogen starts to precipitate in the presence of the antitoxin antibodies. Now, like I said, ELIC test is a gel test. So if we look at this image, I have numbered 1, 2, 3, and 4. The horizontal lines here are going to be strains of bacteria that are going to be tested. And the vertical line here is a strip of filter paper that has antitoxin antibodies on it. Now, you get this filter paper and you line it up with these 4 bacterial strains, and you see which toxin starts to produce precipitation. So as we can see, line 1 and 4 are showing reaction to the antitoxin antibodies on the filter paper strip. So we can conclude that line 1 and 4 bacteria is our diphtheria toxin. But also, anytime this image is shown, we need to understand that this is an ELIC test that is specific for Corynebacterium diphtheria. Okay, so what are the signs and symptoms? So we spoke about lysogeny. So lysogenize or lysogeny is when the normal flora can convert into a pathogen when exposed to a toxin. So Corynebacterium diphtheria would be one of these bacteria that fall under the category of lysogenized organisms. It causes necrosis of your heart, nerve, and epithelial cells. So we see grayish white membranes, also known as pseudomembranous pharyngitis with lymphadenopathy, so a bull's neck appearance. So sometimes the USMLE exams or NBME will actually say an unvaccinated or child has presented to you with gray-white membranes on the throat. And you're supposed to conclude or assume and be able to identify that this is Corynebacterium diphtheria as well. So that's high yield, the gray-white pseudomembranes, because it's not really going to present to you with any other way. This is kind of a gold standard high yield fact here that you need to know, that the gray-white membranes are for Corynebacterium diphtheria. Okay, it causes necrosis of the cardiac cells, so this obviously will lead to myocarditis and arrhythmias. But again, the high yield takeaway here is the gray-white pseudomembranes. So we can give a DPT toxoid vaccine. That is a combination vaccine against 3 infectious diseases seen in humans. So D stands for diphtheria, P stands for pertussis, and T stands for tetanus. So the diphtheria toxin is made by taking the diphtheria toxin and activating it with a chemical. So you take the vaccine, you take the toxin that's in the vaccine, you add a chemical to it, it now becomes a killed vaccine. So the inactivated toxoid or the inactivated toxin is now called a toxoid. So when it's injected, the killed toxin, known as a toxoid, will cause an immune response, but it will not cause a disease. So the patient now has immunity to the toxin. So again, the takeaway from this point is that it falls under the lysogenized bacteria. It causes gray-white pseudomembranes in a bull neck. You have to give a DPT vaccine. And a toxoid means an inactivated or killed vaccine. So other toxic factors that are acquired during lysogeny, and remember we said lysogeny was when the normal flora gets converted into a pathogen. So the mnemonic here for other bugs that cause or that are acquired during lysogeny is COBEDs for cholera, O antigen of salmonella, botulinum toxin seen with Botox, exotoxin of strep pyogenes for E, the D for diphtheria toxin, and S for Shigella toxin. So we got Vibrio cholera, salmonella, Botox, strep pyogenes, diphtheria, and Shigella. So Pseudomonas aeruginosa, the toxin here, is exotoxin A. Exotoxin A also inactivates eukaryotic elongation factor 2. It also contains phospholipase C toxin. This will degrade the cell membrane. It also has endotoxin, which is responsible for fever, shock, and hypotension. It also contains a mucopolysaccharide capsule that forms biofilms. Okay, it contains pyoveridin and pyocyanin, which causes Pseudomonas to have a green and blue pigment color. It's oxidase positive, and that will help it generate reactive oxygen species. Pseudomonas is found in water and it smells like grapes. And this is our gram-negative rod. It's aerobic, it's motile, and it's a non-lactose fermenter. So on this page, the great takeaway here is that it's a non-lactose fermenter. A lot of the times the question will say to you that this bug is a non-lactose fermenter that is oxidase positive. That is the clue that this is Pseudomonas. Now, what are the signs and symptoms of Pseudomonas? So it's in the name, Pseudomonas. P for pneumonia, because it causes pneumonia in cystic fibrosis patients. It does that by the biofilm. The biofilm was your mucopolysaccharide capsule. Sepsis for S, because the endotoxin degrades the cell membrane, causing fever and shock. Ecthyema gangrenosum is the necrotic cutaneous lesions that are highly seen in immunocompromised patients and diabetic patients. It causes UTIs with the endotoxin, and with UTIs we have fever. It's seen in diabetic patients, as I've mentioned, and immunocompromised. This is one of the bugs that's highly specific for otitis externa. So this is your swimmer ear infections. So swimmer's ear infections occurs in the external ear. So this is otitis externa, seen with Pseudomonas, because we said Pseudomonas is found in contaminated water. The M is your mucopolysaccharide capsule, and remember, this is what produces the biofilm. The O is osteomyelitis. The N is your nosocomial, which also means your hospital-acquired infections. This is highly seen in the burn unit. That's another clue. It will also mention that the patient has several burns and they are in the hospital and now they have signs and symptoms of Pseudomonas, or they say that now they have ecthyema gangrenosum because they're obviously immunocompromised because they're on all these steroids from being burnt. So you want to avoid flowers or avoid taking flowers for patients that are in these burn units. And another high yield thing the test likes to mention with Pseudomonas is that their neutrophil count is less than 500. So they have neutropenia with Pseudomonas. So burn units, neutropenia, and ecthyema gangrenosum are 3 high yield points to take away from this slide. A is for addiction in your IV drug abusers. And again, we said the skin infections. So skin infections, because Pseudomonas is found in contaminated water, we will see hot tub folliculitis. Corneal ulcers, when the bacteria gets into the eye, can lead to corneal ulcers. And it can also lead to keratitis in patients who swim in the infected water with their contact lenses. So this can obviously affect your contact lenses and infect the eye. Next thing you know, you have inflammation of the eye. This causes keratitis and ulcers. Next, what is a treatment for Pseudomonas? So the little mnemonic I like here is fire camp. F for fluoroquinolones. Remember that your fluoroquinolones end in floxacin. And the mechanism is that it inhibits topoisomerase 2 and 4. Your 3rd generation and 4th generation cephalosporins are also given for Pseudomonas. Your extended spectrum penicillins is your ticarcillin and piperacillin. So piperacillin and ticarcillin is high yield. Remember that the side effect here is that it causes skin rash. Carbapenems end in penem, and they're given only when there is 100% life-threatening infection. We don't really usually give carbapenems in the US, and it's kind of a last resort drug anyway. So the test probably won't ask you about carbapenems unless it has to do with the side effect. Aminoglycosides inhibits your 30S subunit. Monobactams are also given. They prevent cross-linkage in the cell membrane. Polymyxins like colistin disrupt your cell membrane integrity. And to prevent Pseudomonas infections, you want to avoid flowers and burn units. You want to promptly remove catheters in these patients. Disinfect water-related equipment in hospitals like AC units and things like that, and proper hand washing. So those are your 2 that inhibits elongation factor 2. Now there's 2 more bugs that inactivate the 60S ribosomal subunit by removing adenine from the T-RNA. And these 2 bugs are your Shigella subspecies using Shigatoxin, and your enterohemorrhagic E.coli, which is your EHEC, using a Shiga-like toxin. So the side effect of these 2 toxins are going to be very similar, but let's start with Shigella. So Shigella is only in humans. It's a gram-negative rod, a non-lactose fermenter that's oxidase negative. So oxidase negative, non-lactose fermenter, and it's a gram-negative rod. The transmission is that it will invade the host cell. So it passes from cell to cell, but there's no hematogenous spread. It's H2S negative, which is very important, so I made it bold because we want to be able to identify Shigella from Salmonella. Whereas Salmonella is H2S positive, Shigella is H2S negative. So this contains endotoxin and enterotoxin that will invade the M cells located in your Peyer's patches in the GI system. We want to treat with antibiotics because antibiotics will shorten the duration of infection. And Shigella has a very low inoculation dose that is resistant to gastric acid, and it will cause a disease. Now, a low inoculation dose means not a lot of bacteria or Shigella bacteria is needed in order for it to cause a disease. And on labs, we will see neutrophilia. This will cause neutrophils to increase because it will start to fight off an infection. And we know that neutrophils are our first white blood cells that will be high in count at a site of infection. So neutrophilia greater than 500, greater than 600, and we will know that we have a bacterial infection when those numbers are high. Now, what are the signs and symptoms of Shigella? So one of the important side effect is that you will see first watery diarrhea that will then become bloody. That's how the test likes to ask you about Shigella. It is a patient that comes to you with a diarrhea that was first watery, and then within a few days it has become bloody. I want you to take that point away and put that in your mind and review that over and over and over again, because that is how you will see it asked on a question. And it also causes HUS, known as hemolytic uremic syndrome. That occurs from cytokines being released from Shigella or Shigella toxin. And this causes a triad of anemia, thrombocytopenia, and acute renal failure. Thrombocytopenia means their platelet counts will decline. Anemia means your red blood cell count has decreased. So platelets are low, red blood cells are low, and the patient starts going into renal failure. And we want to treat Shigella with fluoroquinolones. Remember, fluoroquinolones, they end in fluoxetine, and they inhibit topoisomerase 2 and 4. Now let's move on to enterohemorrhagic E. coli, which is our EHEC. So EHEC contains Shigella-like toxin. This also enhances cytokine release. Now, O157H7 is the most common type in the United States of America. So this one, EHEC, does not ferment sorbitol. And that's high yield because other subspecies of E. coli do ferment sorbitol, but EHEC is the only one that does not ferment sorbitol. Okay. It is found in undercooked meat and your raw leafy vegetables, so hamburgers, hemorrhagic, think of the H's together, hemorrhage, bloody diarrhea, undercooked meat, usually hamburgers, and it does not invade the host cell, unlike Shigella. So this one does not invade or enter the actual cell, unlike Shigella, which, remember, invades your M cells. So what are the signs and symptoms of your enterohemorrhagic E. coli? We have HUS, hemolytic uremic syndrome. So the Shigella-like toxin causes a triad of anemia, thrombocytopenia, and acute renal failure. That's due to your microthrombi forming on the endothelium. So now you start forming these little thrombi, and they start blocking these small little arteries, and you lead to renal failure. Dysentery means a bloody diarrhea from the toxin causing necrosis and inflammation. So with EHEC, the Shigella-like toxin, you do see necrosis and inflammation. On the labs, we see platelet consumption. So thrombocytopenia means a very low platelet count. Anemia means low red blood cell count. And what happens is with EHEC, you start seeing schistocytes and helmet cells, because as these red blood cells try to pass where there is a clot or a blockage, these red blood cells start to become lysed. And you start seeing schistocytes, known as helmet cells, which are indication that there is E. coli going on. So the histology is very important because we do see schistocytes and helminth cells a lot later when we go over hematology histology. So just remember here that with enterohemorrhagic E. coli, we do see schistocytes. And that's the end of the chapter review. So I hope you enjoyed this chapter. Take away some of the high yield points that I did mention and try to practice and listen to these slides a few more times, because I can guarantee you what I've mentioned will be asked on the NBME exactly how I've gone over it, and the USMLE exactly how I have reviewed it. The next chapter that we will go over is also your bugs with exotoxin. So look forward to that, and please leave a comment if there is something you need me to go over. Thank you.